Right. Yeah, let's get started then. All right. So our agenda for today is the brief, basically introduction to the ways that we take to analyze the genomes, and then I'll try to give you an idea of what an intelligent genome analysis can be. So I guess so. Let's also take an even further step back and then think about basically the goal of computing that we're doing today, right? So what we are essentially trying to achieve with computing. Well, nowadays, I guess there are many reasons to do computation, I assume, especially, right, for the popular cases, even the entertainment is one reason to do computation. But I guess we should, for now, limit ourselves for scientific purposes. And if we think about those purposes, what we can perhaps tell is that the purpose of computing, at least according to Richard Hamming, which I also agree, the purpose of computing is actually to gain insight, not numbers. Basically, the computers will give us some numbers based on, let's say, the numeric calculations that it's doing, but we don't really want to just see those numbers, right? What we want to do is we want to gain insight. We want to generate answers from these answers from these numbers by doing the computation. So this is essentially perhaps a general definition of the reason of, let's say, scientific computing nowadays. And basically, even today, this is actually even a bigger problem because we need to basically gain insights and observations from these numbers much more efficiently than ever before. So this is because we're generating lots of data today. And this actually, the number of the amount of data that we're generating is increasing day by day. So when you basically think of these use cases such as astronomy or social media like YouTube or X, or even the genomics data that we're interested in this course, is generating lots of data. And this data is actually even perhaps faster than we can process. The basic speed of data generation is faster than the speed of basically the technologies are evolving. And I'm saying this because right now, like, we're even seeing a shift today in the industry from, let's say, from the era of nanometers, right, to the era of angstrom. But this is even smaller than a 1 nanometer, and Intel is actually pushing towards that limit to basically even fit even more transistors to achieve, let's say high performance and even maybe more energy efficient computing. However, we have basically a huge problem. The problem is, so when we think of these important applications like genomics, and as you already know, this is basically an illustration of one of these sequencing machines, like the Illumina machine, right? So these machines, although they are generating really important data, they are essentially special purpose machines for data generation, not to analyze the data, right? And they are really fast to generate that data. And essentially the problem is that when we try to use a general purpose machine to analyze such a data, these machines are really not specifically designed to analyze this specific particular type of data generated from a particular machine, right? So that, and this way they become really slow to analyze this data, and they usually become they are essentially basically bottlenecked by the data movement overheads. So basically, what we have today is a slow and inefficient processing capability that is not capable of larger to process large amount of data moment, right? So, and we're interested in this data because this type of data is usually very important for us. This type of data is usually generated from very important applications, such that they carry really valuable information or knowledge, right? And then we really want to extract that knowledge from this data by essentially achieving certain goals. So what can these goals be? Perhaps maybe there can be a latency constraint or performance constraint, or maybe there could be some energy constraints, or maybe essentially other costs, right, that you can think of to associate with the goals that you want to set for your application. And so basically, such a data analysis or such a processing capability is ideally, or should ideally be tailored for many important applications, such as AI, ML applications, or genomics, medicine, and health. And if you can basically extract such an information extremely efficiently by meeting these goals, we can actually call this type of analysis perhaps intelligent data analysis, because this can meet all of our goals to process the data generated from an important application. And perhaps you're aware, this is actually also one of the key research directions that we're taking in our research group, which is essentially building architectures for important applications such as AI, ML, genomics, medicine, and health. So, but clearly, we need faster, scalable, and accurate genome analysis today for many, many reasons. For example, this type of analysis is needed to understand, let's say, genetic variations, species, and evolution in the population, right? So we really want to understand this variation so that we can understand, let's say, the life scope better, so that we can improve the quality of life by providing clever solutions to it. Another way of analyzing the genome, or why this type of analysis is important, is basically we may also want to predict the presence of relative abundance of microbes in a given sample, right? This can be actually a life-critical analysis if a particular sample perhaps contain really dangerous species, let's say, in the sample. So this is also a bit related to achieve rapid surveillance of disease outbreaks, right? If you have, let's say, our genome analysis setup ready in many applications, and then if you continuously monitor, let's say, such potential outbreaks, then we could actually even take actions much more quickly, right? Even before seeing the symptoms from people and understanding that such a symptom can be dangerous for human health and the health of other living organisms, essentially. And this also an accurate and fast analysis is also important for developing personalized medicine, because as you may imagine, it would be much more efficient and much more, let's say, precise if you could, let's say, prescribe a medicine that's extremely tailored for each individual, right? Such a medicine has a potential to basically improve the quality of life for every individual, let's say, in different ways, tailored for that particular individual. So then I guess understanding a genome analysis can be really important to analyze. Let's then cover the basics of the genome, right? So basically, what is a genome, right? So because I guess everyone has it, right? Otherwise, it literally wouldn't be possible for you to be, I guess, watching this lecture, I guess, thinking about the ideas and thinking about the question, what is a genome, etc., right? All of this possible is because we all carry this let's say, biological structure that's called genome that makes up even other things. So if we want to have a loosey, or let's say a high-level description, a genome is basically an entire set of DNA sequences in a cell, right? So everyone has basically millions, billions of cells, and inside the cells we have essentially DNA. And this is not even just a DNA that's included in your nucleus, which basically is constructing your chromosomes, let's say, but we also have a mitochondrial DNA in our cells. And basically, if we consider the DNA in its digital form or in the human readable form, this is nothing but, let's say, again, loosely, is a series of certain characters, and this character is basically usually in an alphabet of 4 letters, right? If you are considering DNA or not, right? And these 4 letters are essentially ATGCs. Each of them is corresponding to a particular nucleic base or base, right? So adenine, guanine, cytosine, and thymine. And all of these are essentially coming together, makes up the living things, essentially, and tells basic organisms, or provides a structure or the code for organisms so that they know what to do, essentially. So then I guess, like, since this is a series of sequences, you may also wonder, how large is it, right? Does it have a limit? Yeah, sure, it has a limit. Then the question is, how large is it, right? I guess to put perhaps visible comparison, I guess we can think of this particular building. I'm not sure if we are familiar with this building. This is a building in Zürich. This is Andreas Turm, is located in Oerlikon, and actually we have some offices around the top floors here as SAPA research group. And if you really want to compare, let's say, how large a DNA can be, you can perhaps imagine that we write every character in your genome in a letter, and then we keep writing until we finish all your chromosomes, right? So when you do that, what you're going to end up is basically, and also if you place these papers on top of each other, what you're going to end up is that you're going to have a pile of papers that's even longer than the Andreas Turm. That's going to be basically around 100 meters, and the Andreas Turm is around 75 meters, right? And if you are still basically suspecting that if this is true, I did the math here, so you can check and see basically this is true. So this is basically puts a different, let's say, view, right? That's a different concept for us, because DNA or genome is huge. There is a huge amount of data to process, and this is even for a single individual. And this is even just generated once, and usually we generate this type of data many, many times for a single individual, right? Maybe 10 times or 30 times for redundancy purposes. So you just imagine the amount of data that we need to process. And I guess when you then understand how large this is, you can also understand how large could it be to construct basically the complete human reference genome altogether. And this basically has been the effort for the last 30, even 35 years. And the scientists actually started the idea of constructing the full human reference genome in 1990s, and they actually finished this in 2003, around 2003. And this was actually a huge breakthrough back in that time. But this was a huge breakthrough because this took a lot of time and took a lot of money, let's say. basically the institutions spent around $3 billion to construct the human reference genome. And the way that it was done was it almost literally similar to writing every character in your genome one by one to a piece of paper, let's say. It's not exactly that way, but if you want to basically understand the hardness of the problem, just imagine that you need to write almost every character one by one in the human reference genome for every paper, where basically the length of papers are even larger than a tall building. And imagine that you need to do it for every papers to finish up the human reference genome. And this was a huge breakthrough, but the issue, relatively an issue, is that although it was good enough back then, it wasn't complete. We were still missing some parts of the human reference genome. So this was the case up until 2021, when basically as the sequencing technologies improved, it became relatively easier, let's say, or quicker and even cheaper to build up the reference genomes. And so in between the first generation of the reference genome up until today, we also have been generating other reference genomes, and we learned basically how to generate reference genome accurately, faster, and cheaper, let's say. And the tools were, of course, also building and improving within that time. So eventually, as we improved things, we were able to finally construct the full human reference genome, essentially gapless, the complete human reference genome. And this was done by, this was basically an effort of course, many people, but especially led by certain people, as you can see here, especially by Adam Filippi, Evan Eichler, Karen Miga, and Michael Schatz. So these people were also recognized as the 100 most influential people of 2022 by Time magazine last year, because of the completion of this human reference genome. So you can see how high impact this is to complete such a human reference genome, because it's extremely important to do so, to basically understand the human genome better and then to develop tools better. So we keep talking about essentially human reference genome, but let's also consider, to put basically a comparison, let's consider other genomes. So for example, we can start with the viral genomes, right? So viral genomes are essentially usually the smallest, they contain the smallest genomes in terms of the number of bases that they contain. So some of the viruses will contain DNA, some of them will contain RNA as their, let's say, nucleic bases. So let's consider this particular virus. This is 5X174 virus, and the length of the bases that it contains is around 5,000 bases. Right. So this virus can infect bacteria. Right. It can essentially harm bacteria. In particular, we're talking about E. coli bacteria here. So the size of the bacteria in the case of E. coli is around 5.5 million bases. It's almost 100 times, let's say, larger than the viral genome. So E. coli can also be harmful for human, right? So it can be contaminated especially in food samples, and if basically those food samples are containing E. coli, and if you basically are infected by this particular E. coli, this can be extremely harmful for you. And essentially, E. coli can infect humans as well, and humans have around 3.2 billion base characters in their genomes. So I guess there is now a trend. Maybe you can imagine or guess a particular trend here, right? as the genome size increases, maybe the complexity, let's say, of the human genome, of the genome or the life is also increasing, right? So we came from the viral genome, then moved to bacteria, and then moved to human, and the genome size was increasing. So is there essentially a correlation between this? So I would say no, because it's not always the case, basically. So especially when we think about the plant genomes, what we tend to see is that these plant genomes usually contain even longer genomes. For example, an onion can contain around 16 billion bases. So the question is onion more complex structure than a human being? So that's very, I guess, a subjective question, right? So it depends on how you describe the complexity. If you are talking about the intelligence, perhaps no, but maybe there are other complexity levels that we should think of. And even there are even larger plants. There are basically other plants with larger genomes in this. For example, this particular genome, again, a plant genome, contains 150 billion bases, almost 50 times larger than the human genome size. And there are basically many reasons to it. There are evolutionary reasons. Some plant genomes are not necessarily diploid genomes, meaning they won't contain a pair of chromosomes from parents, but they may be polyploid, containing essentially many, let's say, copies of a particular chromosome, leading to increased size of the gene. So having said that, let's also consider this. Let's also cover, I guess, the lower levels of the genome. So I'll stick to human reference genome for now to keep things simpler. So human reference genome contains 23 pairs of chromosomes. One essentially from one parent, the other one is from another parent, basically. And the 22 of these chromosomes coming from one parent are essentially autosomes, meaning they are not correlated with the sex chromosome. So we also have a pair of chromosomes known as a sex chromosome, but these are not called autosomes, but rather sex chromosomes. So this is essentially, in a high level, is a difference. So if you look even more closely, what we're going to see is that this famous double helix structure of the genome, right? So this is essentially a chromosome, and then the structure of it actually will be even more complex than this. So the chromosome won't be basically fully opened, meaning it won't always be make these double helix structure completely wide open, but rather at certain points it will be, let's say, compressed. And for these compressed regions, we call these regions chromatins, and they are essentially compressed by certain structures such as histones that will wrap like a roll. Basically, imagine a roll, and then we're wrapping your essentially string on that roll. So those are essentially histones in the genome. But for the other parts that are, let's say, not compressed and wide open, we can basically see the open structure of this double helix structure of the DNA in this particular case, and those will basically will also contain certain bases or nucleic bases that we call adenine, thymine, cytosine, and guanine, essentially, and they will basically bind to each other based on the chemical interactions between them. And as you may know, adenine will bind to thymine, and cytosine will bind to guanine, and so on. From basically, in one strand, if you have an adenine, then we can perhaps, with a very high confidence, say that we have thymine in the other strand. So this is basically how, in reality, it looks like under electron microscope. So this is basically essentially a pair of chromosomes. So this is basically the centromere region, and these regions are telomeres. in the chromosome, basically the end joints of the chromosomes, which are particularly hard to, let's say, construct for, I guess, for let's say more low-level reasons. But essentially, this chromosome will be, in most cases, will be compressed, right? Meaning, you won't really see the DNA wide open here, except for particular regions. So then, I guess we can also then try to answer how that DNA is useful in real life, right? So I guess we can answer this quickly by describing the central dogma of let's say life or molecular biology, right? So we have DNAs that it consists of ATGCs and so on, right? In both strands. So we have 2 strands and 1 strand, and there's a second strand in DNA. So you can think of, if you want to make an analogy, you can think of this DNA information as a source code in your, let's say, in the program that you're perhaps in the code that you're writing. And this source code will not always carry useful information, let's say. Right? Sometimes it will contain comments, which is useful for you, but not really useful for the machine, right? It will contain macros, I guess. It will disable that particular source code depending on the flags that it receives, right? So it has this particular functionality, but essentially what it tells us, it basically defines the functionality. Doesn't perform the functionality, but it defines the functionality. But this is also perhaps the role of DNA. It's defining the source code. Let's say it's defining the functionality. And what happens is that we can transcribe this to an RNA. So the RNA is essentially going to be generated from the useful regions of the DNA, extremely similar to what would happen if you converted your source code to an assembly or a binary, right? Your source code, basically your comments or macros, let's say disabled macros, will not be carried out to the binary, right? Only the useful part will be carried out to the binary. But this is also similar in the case of RNA. So this essentially will contain the useful part. So then what happens is that, so once we transcribe the DNA to RNA from the useful parts of the DNA, the other reactions will take into place, and then this particular RNA will be converted into a series of amino acids making up the protein structure. And this essentially is what's, let's say, regulating, or what's really functioning, let's say, in the cell. And this is also similar to how your binary would be executed on our processor, right? It's now like we have the binary useful part, but let's now put it into it's basically is telling how the processor should work, I guess, right? So this is now executing something and doing something. It's basically function. So this is the case of also the protein. Also, is what's making up the real functions, such as the phenotypes. For example, your eye color, your hair color, or essentially how your particular cell regulates itself, and so on. So I hope this was clear so far, because we're going to also delve into a little bit more detail about, let's say, what's making each cell different, I guess, right? Because essentially the idea is that the DNA or the genome that we carry in our body will be contained almost in exactly the same way in our cells, right? So this means that most cells in our body will have almost the same DNA and the same, let's say, genes. But then, if that's the case, like how come we have a skin cell and also a liver cell and perhaps, I don't know, like a muscle cell and the other things, right? So how do they differ than each other? So this is also, so I'm not going to go back into that lower level. This is essentially initially defined by the signaling early on the development of the tissue or the organism, basically in the single cell process or a multi-cell process. So when you're considering the human, this is basically happening in the early days of the of basically when you are just basically a particle, like a part of a set of cells in your mother's, let's say, body, right? So this type of signaling in the early days of life will essentially affect how genes are going to be expressed in each cell. So as you may remember, I told you that like the DNA is not always fully open. It's in certain places it's compressed, in certain places it's open, etc. So this also is affecting how each cell is behaving differently. So it's not at certain places, in some particular cell, like some parts of the DNA will be open, but when you look at in other type of cell, you'll perhaps see that that part is closed. So this will essentially affect how the genes are essentially opened, like switched on or switched off in each cell. And these genes are telling which proteins to generate. And since we can only use the switched on, let's say, genes, we can only generate proteins from these parts of the DNA. So then this literally means that once you compress and uncompress, and for like once you have like other chemical bindings in your DNA, so I'm just making this a little bit simpler. So this is basically telling which genes are on and which genes are off. And knowing that, we can also tell, okay, then in this particular cell, we're going to be perhaps generating this particular RNA, and this particular RNA will lead to this particular protein. It will then lead to this particular type of functionality, and so on, essentially defining the role of each cell, making up. And then once these cells are close to each other, the signaling between these cells will also affect the functionality and so on. So this is essentially how each cell can differ from each other. Right? And also over time, mutations can occur. We call them somatic mutations, that the ones that are occurring over time. So this may also affect the functionality of the gene. Right? So, for example, if a particular mutation occurs on your particular location in your gene, then maybe this can switch off that gene, disabling perhaps even synthesis of a particular protein, maybe leading to a particular disease, and so on. So I guess having said that, you perhaps realize it's really important to understand the differences, let's say, between a healthy cell and also a cell that shows a particular phenotype, let's say a particular function. Maybe, let's say in this case, a blood pressure. So perhaps like when you look at the individuals that have relatively low level of blood pressure, you may categorize a particular region, saying that if you have a low blood pressure, then you're likely to have this type of information in your DNA, meaning perhaps you'll always see T over here if you have a relatively lower level of blood pressure. But maybe due to a particular mutation or maybe the inheritance from your parents, maybe you don't have a T but C over there in one place of your genome, right? And having that may have a potential to show a particular phenotype. Again, in this case, the blood pressure. Maybe this can lead to increased blood pressure just because you have a particular change at a position in your genome. And we call these changes SNPs or single nucleotide polymorphism. So this is essentially telling there is a single change in the nucleic base compared to some other, let's say, controlled genome. So this is another example. So we may be doing essentially a genome-wide association studies. This means that you may gather a group of individuals where they may be relatively healthy, let's say not showing a particular symptom, but then you may also gather a group of individuals that like showing a particular symptom. And what you can do is that you can sequence the genome of all of these individuals and then try to find a correlation, basically, between the synapse and the particular symptom that these individuals are showing, right? So perhaps maybe particular, so it doesn't mean that whenever there's a synapse, it will be basically linked to that particular symptom, because maybe it may be linked to something else, or maybe it may not be linked to anything at all, right? But when you have this control group from many individuals, and then if you keep seeing, let's say, a high level of, let's say, evidence that the particular synapse or particular mutations are occurring in this particular group of individuals, but not occurring in this particular group of individuals, then perhaps you can link that synapse to that symptom, saying that, oh, okay, like if you have this synapse or if you have this mutation, then you're likely to have or to show that particular symptom. So as you can see, this is mainly statistical based on the observations. And if you are interested, you can check and see this paper to understand how this type of study is carried out. And we're also building, let's say, dictionaries that will literally tell you if you have this particular synapse, meaning if you have this base, let's say, in a chromosome at this particular position in your genome, and if this is a synapse, meaning this is not really observed, let's say, in the general population, you can basically check, like there are names for that particular synapse, and then you can go and check the dictionaries and then see what that synapse having, what could it mean, basically, having that particular synapse in your genome. So it may tell you that, oh, like with this particular percentage, you're likely to have this particular symptom, and so on. So we're building this day by day. So and this is basically an example from an OpenSNP database. So we don't also just have single mutations. We also have larger mutations, meaning maybe a huge portion of your, let's say, genome may be deleted, right? Or maybe it could be inverted or like moved to somewhere else, and so on. So you may imagine that such large mutation may also lead to particular symptoms, and this is in fact the case, understanding, and these variations are usually called structural variations, and understanding these structural variations are very important to understand, let's say, the diseases may be linked to the structural variations. And there are some examples here, for example, autism, schizophrenia, obesity, and having been underweight are essentially linked to particular structural variations in your genome. Yeah, I mean, if you are interested, you can also check this paper. So then I guess let's go into the second part of the lecture today, where we'll cover the intelligent genome analysis. So we've loosely mentioned the intelligent genome analysis in the beginning of the lecture, right? We had some goals and we wanted to achieve that, and so on. The question is, and does intelligent genome analysis really matter, let's say? Well, then I guess it depends on what we really want to achieve from intelligent genome analysis. So there are some, in a broad level, there are some, I guess, goals that can be set. For example, we may want to achieve fast genome analysis, right? This may be related to the latency or the throughput of the analysis, or both of them together. For example, you may want to do some real-time analysis as the sequencing data is generated. So this also should, such analysis can be large scale, should be applicable to large scale, right? Meaning you may want to analyze the entire population, and doing so, you don't really want to slow down your analysis a lot. So you just want to have a good scalability that can make it applicable to the larger genomes. And we also want to, of course, have an accurate analysis, right? This is important for precision and accuracy reasons. You don't really want to do inaccurate diagnosis of the disease. In a sense, you also want to use the right architectures to do our analysis. Doesn't mean that we always want to rely on the general purpose architectures, but perhaps there are better fitting architectures out there that we can use to achieve fast and accurate analysis, like by meeting the energy efficiency constraints and the bandwidth constraints. Yeah, and the other, I guess, criteria is the privacy. We all know that the DNA is a very valuable and sensitive asset that we really want to protect. So that perhaps should be also protected, and then to make sure the safety of each individual. And we describe these also steps in this paper. If you are interested in learning more, you can check this paper. So let's go over, I guess, one by one very quickly. We can start with the fast genome analysis, perhaps. So what we mean by fast genome analysis, I guess, in general, perhaps fast genome analysis should be done in mere seconds, using maybe even limited computational resources, for example, personal computer or a mobile device. So this is basically one example of fast genome analysis. And we want to do this because such type of decision or analysis can be really life critical. For example, so this is actually coming down in the UK right now. If we have basically critically ill infants and newborns, it's essentially required to have a whole genome analysis of these infants. And this is actually also known as rapid whole genome sequencing, or RWGS. Whole genome sequencing would mean you're sequencing the entire genome. And this essentially means that we want to do this analysis extremely quickly, of course, because this is life critical, and then some answers should be given extremely fast. And as I said, from 2019 and on, all seriously ill children in the UK will be offered a whole genome sequencing as part of their care. This is extremely important to understand certain issues or diseases for certain illnesses, right, to diagnose them quickly. And also, this is useful to sequencing or genome analysis is useful for rapid surveillance of disease outbreaks. We can essentially use it, for example, to Zika virus to avoid its spread in a certain community. And this was actually done and used previously in 2017 using nanopore sequencing. So this is another example, essentially, for Ebola virus, and this is finally done for also the COVID-19. So if you remember, I guess, during the COVID-19 outbreak, it was extremely common to do PCR testing. So PCR testing is, of course, extremely reliable. It's providing us extremely reliable results, but this is only possible for non-regions to target. So if you don't know the region to target, then you cannot really design a PCR. So you should first know which regions to target, so that you can design your PCR. So this means that there is actually a high latency to get the right answer, and it is hard to customize to any region, let's say. So I'm saying high latency because when you give the sample, there's usually a few hours that you need to wait to get your answer. And ideally, this can also be done. This could be done using sequencing techniques. Again, in this particular example, nanopore sequencing, so that you can give answers, basically, whether a particular sample is contaminated by COVID virus or not, doing some DNA sequencing. So the other aspect was the large-scale analysis, if you may remember. So our goal in this large-scale analysis is basically to understand, for example, what organisms are present in a given environment and how abundant they are. This is important, essentially, to understand the other living cells that are essentially living with us, right? And it's important to understand them so that maybe some of these microbiomes, some of these organisms may be harmful for the human health. The other way of doing this large-scale analysis is a population scale analysis, right? So we may essentially take a particular population. In this case, in this particular example, we take, for example, 50,000 individuals, and we want to sequence their DNA and then analyze their genome. And in this case, such an analysis, population scale analysis, takes 83 CPU hours per sample. So if you multiply this by 50,000, this is equivalent to 4.50 million CPU hours. So this means that, like, for example, if you have a server with perhaps CPUs of size, let's say 84 CPUs or 64 CPUs, this is roughly taking 1 hour, let's say, per sample. And even in such a server, it will take 50,000 hours to get the entire answer, right? And we're even talking about this such a large scale CPU to do such a population scale genomics. So you can clearly see the need to develop faster and accurate tools to analyze the genomes in a scalable way, so that we can get the answers from everyone extremely quickly. There are also some efforts essentially to build and profile viral genomes, and also industries are coming into play. For example, RWAs is offering their compute capabilities to perform such computation in large scale. We may also perform city-scale microbiome profiling. This means that we may collect samples from many regions in a city, in this case, for example, New York City, and perhaps you collect samples from, let's say, a few thousand places. So this means that you actually need to, again, sequence such a large sample and understand it, whether it contains a harmful organism or not. So essentially, I guess, like, having understanding and a large scale analysis is important. The other point is that the accurate analysis is also extremely important, right? Because we're making life critical decisions. And this is also important to improve the quality of life. So this is an example, perhaps, why an accurate analysis is important. So there was essentially a question of whether there's a plague in the New York City, New York subway system. So plague is, as you may know, has killed essentially millions of people in Europe in the 14th century. It's also known as the Black Death back in that time, and is essentially extremely harmful for the human lives. And once we were doing this city-scale genome analysis in New York City, what researchers thought they found is that there could be samples of plague in the New York City sample, and this, of course, raised an extreme concern for the people living in the New York City. But essentially, this people realized that this was essentially a false information, not essentially an incorrect analysis. And this was essentially referred as a failure of bioinformatics. But essentially, this only means that we just need to develop tools that are scalable and also accurate to give the right answer and also quickly, right? So perhaps back in the day, we didn't have such tools that are scalable and also accurate to give these answers. And the issue essentially was another sample that is not that harmful was essentially containing a similar DNA strand, and then that's why people actually were confused and essentially thought that this particular DNA region may be coming from a flake and not other genome. And to improve these tools, of course, right now we have benchmarking suites and also almost yearly done benchmarking analysis. So this is basically a critical assessment of metagenome interpretation analysis, or CAMI. And this is essentially like every year, people are submitting their tools to that competition, let's say, without knowing the sample. And the people are testing each tool with the sample that they collect, and then ranking each tool based on their accuracy, performance, and so on, so that people can keep improving these tools such that we'll have a fast and accurate analysis. So the other part of, I guess, the intelligent analysis is to use the right architecture for performance and also reliability reasons, right? So, so this is basically maybe non-genomics related context, but you may think that perhaps when you're on a plane, you really don't want the plane, let's say, to have a failure, right? To have a reliability issues. But it may happen essentially. And our goal is to prevent that from happening. And this is essentially also the case in the genomic studies. We may be trying to analyze the genome in many different environments, including the other space. Right? This is an example of sample preparation and DNA sequencing, and in the other space. So it doesn't even mean that we're going to be analyzing the genome on Earth, but we may also want to analyze it in the space and even on Mars. So this is an example of the preparation, let's say, of how we're getting prepared, basically, to collect samples from Mars and also to sequence the samples that we collect from Mars, even on site, meaning on Mars. So there is an interesting read, which was published a month ago. So if you're interested, you can read this. But of course, the other question is we also want to use intelligent architectures, right? So we don't really want to, let's say, always use a general purpose machines, but find the right machine or find the right architecture to process our data extremely fast and accurately. Right? In this example, maybe we don't have perhaps a server size machine, or maybe we just have a mobile device with limited resource capabilities, right? And the question is, how can we co-design the architecture and algorithm so that we can do our analysis on the mobile device? I guess the last point is to remember is DNA is a valuable asset to protect. And this has become even more important today because you may be aware there was this recent data leakage from 23andMe company. And 23andMe is essentially what they are doing. You're sending your DNA sample, and then they are analyzing, they are sequencing and analyzing for you. But the question is, what happens with that data, right? So now they have your data, they have your, perhaps the most valuable information, right? And in this case, even worse, if this data is leaked, for, I guess, data is leaked to the individuals that have, let's say, not so good intentions. That's even more perhaps scary and dangerous. And in this particular case, this particular study published in Washington Post is claiming that after a hack, it appeared that these hackers were targeting people with Jewish ancestry, right? So people are now wondering how to cut ties with the company, right? So that their data is not leaked, even if it is not leaked even now. They want to prevent that this data leakage, so that they won't perhaps be targeted in the future. And of course, there are some measurements that we're taking. There are some privacy preserving genome analysis that takes this issue into account, tries to protect your most valuable information. And basically, if you're interested, you can check this paper, and even there are many other papers taking this into account. And now even the companies are proposing or promising that they perform the DNA testing by taking the privacy into account. So they are saying that we essentially don't release or make your data private during our analysis, so that even if it is leaked, hopefully it won't be visible to the hackers. So I guess this way you can understand faster, scalable, and accurate genome analysis is extremely important for many reasons. And this is not even true for just genome analysis. This is true for many, many applications. And I guess the applications are only limited by our imagination right now. So for example, genome editing. Right now, we can pinpoint a particular bases in your DNA, and we can, let's say, cut those bases in your DNA so that you can switch off the functionalities that you don't want, or even maybe switch on certain functionalities. There are newer technologies that will that let us to insert the bases that we want in the genome, so that we can have the particular functionality. So this is extremely interesting, extremely exciting, because it has a tremendous opportunity to lead up to significant things. And since this is extremely interesting to do so, and this is programmable essentially, right? So you can literally program almost to change almost any region in your genome. I'm saying almost, there are some conditions to do it, but let's say the fact that this is programmable, this is extremely interesting. And since, because of this, basically the opportunity, the Nobel Prize was given to these two individuals, Jennifer Doudna and Emmanuel Charpentier, got Nobel Prize award in 2020. And this was actually the product of the paper that was released in 2011 or 2012. So imagine having the Nobel Prize in 8 years, just because of how exciting this is, right? How it can lead to bigger things, right? So this is basically very interesting. So other interesting perhaps point is the DNA computing. So we don't now just use these digital machines or analog machines to perform computation for us. We can now also use DNAs to provide some answers for some hard problems, let's say. So in this case, you may imagine a traveling salesman problem, right? So you have connections between cities, and then you're wondering, when you start from a particular state, can you end up, can you basically visit every state, like maybe even once, and then end up in any other state? And what people are now doing is that they are actually encoding each city with a particular DNA strand, such that the connections between cities are also encoded at the end positions of these DNAs, so that we can use the chemical structures. Meaning, for example, when you have an DNA encoding for Los Angeles and Dallas, they'll have this chemical bond over here, so that they can connect to each other, because in our digital representation of this graph, they have a connection. And what you're wondering is basically, you have this graph, but you don't know the answer, whether there's such a path, basically, right? From one place to another place. And this is actually an NP hard problem. And what people, meaning, if you have, let's say, unlimited number of parallelism, perhaps you could find it in the polynomial time, but in reality, you don't have such a parallelism. So what we're trying to do is that since DNA is offering us this huge level of parallelism, perhaps like millions of parallel computations that can happen at a time, what people are doing is they are encoding the cities, representing the graph, and then letting the DNA work does its job. And essentially, if there is such a path, what will happen is that these cities will connect to each other with the chemical bonds. So not all of the DNA strands will connect to each other, but only the ones that is supposed to be connected to each other. And once you sequence, if you see a path from one position to another position, then maybe you can answer your Taylor-Salesman problem quite quickly. Perhaps more quickly than you would answer with a computer, a regular computer, let's say. There are also like other paradigms. For example, you can even shop with your DNA nowadays. So basically, I guess to wrap up, achieving intelligent genome analysis is knowing how and where to enable fast and accurate, cheap, privacy-preserving, and exabyte scale analysis of genomic data. So essentially, this is what we are trying to achieve in general. And this has been our dream, basically, since this was the dream also of the Safari Research Group. And this is basically, the idea is that building an embedded device that can perform comprehensive genome analysis in real time, maybe within a minute, right? To answer some important questions. And to achieve this, we should really think of the entire stack of the computer architecture, from the problem part, perhaps even to the algorithms, even down to the electrons. So we're not really, perhaps for now, very interested in electrons, perhaps even interested in this part. But what's really important is to design all these steps over here together so that we can co-design algorithm and the hardware together to answer our questions quickly and efficiently. And if you're interested in basically learning more in this topic in the context of genome analysis, this is our paper that was presented in DAC this year. Our paper is also available online. And also, we know that there's a bright future for intelligent genome analysis because the sequencing machines are improving. We are designing intelligent architectures. So it's almost a matter of time, let's say, to perhaps even achieve our goal. And I guess to give you a context of the steps in like how genome analysis is done in real time. What we do is usually we collect the sample, right? Maybe it's a blood sample, it's a sample from somewhere in the city, etc. But we collect it, you prepare the sample, you purify it, you amplify it. Essentially, this is a part of DNA sequencing, and you essentially what you do is that we have then the fragments of the DNA that the sequencing machine takes, such that it generates some data. So this data is not directly ATGCs, but it's rather some raw sequencing data. So such a data can be electrical signals, or it can be a set of images or a movie, depending on how the machine is designed to work, right? But the goal is basically to understand that raw sequencing data and maybe either translate it to the basis or even without translating to the basis, perform some computational analysis to understand the genome. And people are developing many tools for many steps in the genome analysis pipeline to achieve fast and accurate genome analysis. And this is essentially the general pipeline of a regular genome analysis. It starts with sequencing, collecting the data, translating the signal perhaps optionally to the human readable form, meaning ATGCs, and then identifying some differences and similarities between sequencing data and some known genome of a species, for example, reference genome, and again, figuring out what the differences are based on your analysis. And next week, we're going to be starting with the first step, obtaining the sequencing data, how the sequencing machines work, and so on. And in the coming weeks, we're going to be covering the other steps. So with that, this is basically the end of the lecture. I'll quickly check in case there are any questions on YouTube. If there are, I'll try to answer them. So I think there is one question by Stanislav. I'm sorry if I saw this question a little bit late, but if you are still around, I'll answer it. Yes, so the question is, the whole genome sequencing, does it mean analysis of all 3.2 billion bases? So in the case of human, yes. So the whole genome sequencing literally means that whatever the organism that you want to work on, let's say it's a bacteria, it's a human, it's zebrafish, something else, you are interested in getting the information from the entire genome, meaning from all chromosomes, even perhaps including the mitochondrial DNA. There are actually several types of sequencing, but there's another type of, let's say, sequencing, which is known as whole exome sequencing. So in this case, you are actually interested in the parts that are known as exons. So those exons they make up, let's say, genes that are particularly responsible for how the proteins are, let's say, synthesized and generated. So if you are just interested in basically the gene locations, there are some other preparation mechanisms that will literally, let's say, delete everywhere in your genome, but the surrounding regions of these exons, so that you can only sequence that part. So this is also known as whole exome sequence. I hope I was able to answer your question. And I don't see any other questions. So with that, I'll finish the lecture. So thanks everyone. I'll see you next week.